Our next application of biotechnology is southern blotting. Southern blotting means that you're taking a piece of DNA that you know is complementary to something of interest that you're going for, and you're using it sort of like this fishing example we had earlier. You're labeling that piece of DNA with a fluorescent label, and then you're going after finding your piece of interest. One application to that is RFLPs. That's basically genetic fingerprinting. RFLP stands for Restriction Fragment Length Polymorphisms. And basically, you're taking some DNA, let's say on a crime scene, you found some DNA from a person, and you're cutting the DNA, and where these restriction enzymes are making their cuts is unique to a person, and it generates fragments that are establishing a genetic fingerprint that is unique to every person. So basically, restriction fragment length polymorphisms means that you are taking some DNA, you're cutting it with the enzymes, and then you're separating the fragments that you generated with gel electrophoresis. Then you're coming in with your fluorescently labeled probe and looking for a piece of interest. So here is an example for this. Your gene of interest is this little piece right there. And now you're cutting up the DNA. So here's this whole blob of DNA, and you're going to cut it up into a bunch of pieces, and then you will separate these pieces by size in a agarose gel, and you can see these bands right here. So these bands patterns that were established, that's all these DNA pieces that were separated by size. The larger pieces are staying closer to the top, and the smaller pieces migrate faster in the gel. And then you will transfer these bands that were generated onto nitrocellulose using the setup right here, where you're basically using capillary action to suck the contents of the gel onto a piece of nitrocellulose membrane. So the gel is down here, and then the nitrocellulose filter sits on top of it, and you're using the sponge and paper towels to basically suck the DNA from the gel out of the gel into the nitrocellulose membrane. And you're peeling off your nitrocellulose membrane, and you can see the pattern of bands that's now here on this nitrocellulose where they previously were in the gel. They still show them in the gel, but anyway. So then you're coming in with your fluorescently labeled probe right here, and it will bind to its matching sequence by complementary base pairing. You have that right here, and then you can get your pattern of bands that your labeled probe found, and compare that to some other pieces of DNA that you cut up in a similar way. So restriction fragment length polymorphism, that's one application of southern blotting, but southern blotting is a very general method of going after complementary DNA pieces using a labeled DNA probe. So here, the application was forensic microbiology right here, DNA fingerprinting. You can use that to identify pathogens. So you will basically cut the DNA from an organism up into many pieces, and then look for pieces that are specific to, let's say, E. coli or Staph aureus or whatnot to identify pathogens. So that's one way. PCR microarrays and DNA chips, they can also screen for multiple pathogens. That is a fairly new technology. DNA chips, they've been around for a couple decades, I guess now. When I left UCI, they were just starting that, and that was in the year 2001. Long time ago. Anyway, so DNA chips, they basically do PCR reactions on, let's say, 10,000 genes simultaneously, or let it just be 100 genes simultaneously. At least you can screen for multiple, the presence for multiple pieces of genetic information, and then find out whether a certain pathogen is present or not present in a sample. And here would be an example of a DNA fingerprinting. And it's using, well, the goal here is to track an infectious disease. So here basically we have an infectious pathogenic E. coli, and you're trying to track down the changes between a non-pathogenic and a pathogenic E. coli and see where the change happened. And so here you have several E. coli isolates from patients who drank contaminated orange juice or some other juice. And so here you can see the pattern of bands, and these are all look pretty identical here. Apple juice isolates, that's the apple juice, and it all looks pretty similar right here. And then here, isolates from patients whose infections were not juice related. You can see that there are quite a few differences right here. Nanotechnology, that's the term that you would use when you're saying that you're manipulating something really, really small, like a molecule. So you're manipulating some DNA, for example, or some other molecule. And here's some example for that. Some bacillus cells that are growing on selenium, forming chains of elemental selenium. It's I don't have much more to say about it here. This is interesting. Agricultural applications. The TI plasmid, that one occurs in Agrobacterium tumefaciens, and it integrates into the plant genome and causes tumor-like growth. So what happens with the TI plasmid is that you can basically use it as a vehicle. And so you're using the TI plasmid, which you can manipulate, and then you're putting the plasmid into the bacterium, then the bacterium infects the plant, and then inserts the plasmid and all of the genetic information that is found on the plasmid into the plant, and then it can basically lead to genetically modified plants. So you can use the TI plasmid and Agrobacterium tumefaciens to introduce a recombinant DNA into a plant. And here's some examples for that. Here would be the tumor that the Agrobacterium tumefaciens can induce, seen here on a rose. And here would be the procedure, the general procedure using the TI plasmid as a vector for genetic modification plants. You start out with your TI plasmid right here. You're going to manipulate the plasmid. You're going to put a new foreign DNA piece into the plasmid, so you're making a recombinant TI plasmid out of that. Then you put the plasmid back into the bacterium, the Agrobacterium tumefaciens, and then you let that bacterium loose on that plant, and now the DNA that was found on the plasmid is being inserted into the DNA of the plant, and now you are having a genetically modified plant that is growing. This is still, what was it previously, a strawberry plant? It's still a strawberry, but it can now maybe do new tricks and can do new things. Sometimes these things are used to make plants resistant to certain pests or fungi. Sometimes they make the plant resistant to pesticides such as or herbicides like the Roundup. So there's different ways you can do this. But Agrobacterium tumefaciens has been used for a long time to genetically modify plants. Here are some agricultural applications of where the Agrobacterium and the TI plasmid that was modified had been used. Very pretty popular, and it's the Bt toxin, and then herbicide resistance, that's usually the Roundup resistance, and then suppression of genes like antisense DNA usage, or for nutritional purpose to make something more nutritional. That's actually true. For example, the golden rice is a good example for that, where rice was engineered to contain beta carotene, so that kids that only live on a bowl of rice per day, they have the beta carotene, which they can then convert into vitamin A, and they need that for vision, to have the rhodopsin that can be made in the eyes. Also, human proteins, you can have them produced in agricultural products. Here are some examples shown of agricultural products. The Bt cotton and Bt corn, those are pretty popular products right here. Genetically modified tomatoes, raspberries, the Pseudomonas fluorescens bacterium. You may have heard of the flavor saver tomatoes, some genetically modified raspberries. Almost a vast majority of the corn in the United States and soy is actually produced with using biotechnology and using the some sort of basically genetically modified corn and soy. And then here are some other examples. The glyphosate resistance, that's the big story, the Roundup Ready story here. And these plants, they have the bacterial gene that allows the use of herbicide, Roundup, weed killer, without damaging the crops. And this is the vast majority of the corn and the soy that's in the United States grown right now. There are some other products that are listed here: the bovine growth hormone, porcine growth hormone, and transgenic animals. They're done with with this biotechnology, and then other food production products, cellulase, chymogen. Well, anyway, you may have seen on milk labels that something was not produced with the use of RBST, the bovine somatotropin, the bovine growth hormone, basically, that is. This is right here. So they have a recombinant version of the bovine growth hormone that was injected into cows to have them produce more milk. And a lot of people didn't like that for good reasons and just kind of started refusing to purchase those products. And nowadays it's a label of, yeah, we're not using the RBST for our products, and we don't buy milk from farmers that are using this RBST. So it kind of backfired a bit and for some of the products. There's some safety issues and ethics of using DNA technology. Well, depending on what the gene is and what it does, there really needs to be some sort of a control that it's not accidentally released into the environment. It's just sometimes hard to do because these genes, they replicate and they're sometimes being carried on from crops to crops. And then also, this is a big issue lately, that these genetically modified crops, they must be safe for consumption and for the environment. And when it comes to the Roundup Ready soy and corn, those tests that were done, they were done very briefly and very sloppily. And so these products got approved for use very fast. And there's a lot of backlash now in California. There were the first lawsuits going on that people said that they got cancer because of because they were using or eating the Roundup Ready corn and soy products, so genetically modified organisms. So there's quite a bit of backlash, and they should have done a better job in testing that these genetically modified crops are actually safe. And then when it comes to individual genetic information, that's a whole different issue here. Who's going to have access to somebody's genetic information? The databases are growing, and a lot of people are not even concerned that their 23andMe DNA is out there, and it's basically public. So I'm not sure, but there would be a whole big discussion about who will have access to genetic information and privacy rights. Now let's finish this chapter here with taking a look at the clinical focus. And originally in the Tortora, they have the Norovirus outbreak as an example here for clinical focus. And so the question was asked, so who is responsible for the outbreak? Well, right now, everybody's talking about coronavirus. Nobody was even worried about norovirus. So the question, of course, it started all in China. The question is, of course, these outbreaks, are they related? And for the coronavirus, we clearly know that, well, the outbreak that was first in China and then the focus shifted to Europe and now in the United States. So the source where it started, that seems to be pretty clear. There are other questions that people are asking, though. Is that virus mutating? And for that, you need to monitor the change in the genetic material. And that's where this here comes in, and that's where it fits into the biotechnology side. So basically, with something like the coronavirus outbreak, you're going to monitor the genetics of the virus. Now, it's an RNA virus, as you probably know. And so there, you need to monitor the genetics of this virus and see how it's changing. And so here, they're just doing some epidemiology, which is going to be our next chapter, by the way. But they're looking at case numbers reported and where they were reported. And that's the kind of stuff that you hear in the news all the time right now, where there are outbreaks, where is the focus of an outbreak. And so biotechnology can really help to track something back. Here the example is given that they're using RTs, reverse transcriptase, PCR, polymerase chain reaction, to track down sort of the origin of an outbreak. And then here you can see that you can use restriction fragment length polymorphisms to track down genetic changes. Nowadays, mostly everything's done with full sequence analysis. And so, biotechnology is very useful, especially sequencing and PCR reaction, very useful for analyzing outbreaks.